So I'm going to be talking to you a little bit about nutrigenetics, nutrigenomics, what it has to do with nutrition, and try to introduce why we need to think about moving there in the near future. No special conflicts of interest. So it's my belief that what limits nutrition research is the amount of noise generated in our experiments. We have large variances in our data, and I will put to you that the inclusion of that noise explains why some people find something 1 week and 3 weeks later find something else, and why many people put up major review of the literature manuscripts, that meta-analyses that then say there's nothing to nutrition or diet supplements, it just doesn't work. And the reason is there is a misunderstanding about what we are metabolically. So right now, most people think of us as all the same, varying around a bell-shaped curve, and that we're either all going to respond to something or we're not. Now, the truth is much more likely that there are responders and non-responders. And there may not be 2 curves, there may be 5 curves of different types of responders and non-responders. And the more we do research in nutrition and metabolism, the more we realize that people differ quite widely from one another. And the big question, and the question that my research institute at the University of North Carolina focuses on, and I think that the next big frontier in nutrition is how can we explain our metabolic nutrition individuality. And many of you in companies have been thinking about individualized nutrition and how would you offer products. But I want to point out something about how this individuality and how understanding it could help us at the research end as well as at offering different products to people so that we could understand what works and doesn't work better. And what does cause this variation? There's going to be a number of things. Clearly genetics, I'm going to focus on that today, but epigenetics, which is the marks that we put on genes and the controlling areas for genes that turn the genes on and off, is going to be important, and that's many of you have been thinking about in terms of explaining why early life exposures changes our metabolism and whatever. I'm not going to spend any time talking about that today, but it's an area that's important. A lot of people have been very excited about the microbiome and that we have different bacteria in our intestines, and they may be causing us to be different from one another. But truthfully, diet makes us different because, for instance, we know that certain nutrients turn on P450 or CYP enzymes in our liver that turn them up or down, so that if we've been eating something in the diet that turns those genes on, then the next things that follow may be metabolized more or less rapidly, making us different from each other, at least for the short term, after we've been on a different diet. Our body composition, our environment, many other reasons. But today, I'm going to focus a little bit on genetics and genomics and how you could practically use it in the future. So genetic variation, single nucleotide polymorphisms. There are more than 2 million of these genetic spelling errors that make people different from one another, and each of us has approximately 50,000 of these differences. So though we have discovered the sequence for the human genome, we really haven't discovered the sequence for your genome, because you could have 50,000 discrete differences from what we consider to be the quote, normal human genome, and I'll talk to you more about that later. So these are single nucleotide, single codon substitutions. If they're in a place that doesn't have much to do with metabolism, they don't do much. But if you happen to have one that's right in a gene that's controlling how you metabolize sugar or somewhere else, it might be very important at readjusting or changing your metabolic pathways. So if you think about it, if we have a gene that makes an enzyme, a protein that has to convert precursor to product, and that gene has a substitution that results in a different amino acid being incorporated in it because the gene, in the protein it encodes for, that protein may fold slightly wrong, it may work slightly less well. I guess it's possible it might work even better. So you could have a decrease or an increase in function. Let's say you most likely have a decrease in function. You might also have a single nucleotide polymorphism in the area that switches that gene on and off. And if it can't be switched on and off, then the gene, even though it's perfectly normal, can't make the product it's supposed to make to drive metabolism. So often, we will have inefficiencies, metabolic inefficiencies that are caused by the fact that we have been blessed with a genetic code that makes a junky enzyme. Now, I want to stress that many people here have been familiar with genome-wide association studies, and many have been frustrated at the fact that very little has come out of genome-wide association studies. And there's two reasons for that. One, people are asking too many questions at the same time, and in science, when we ask too many questions, we have to make too many statistical corrections. And so unless you have a huge power, if you ask 2 million questions, you'll never get a good answer unless you have tens of millions of people to study. And that just has to do with statistical power. You just can't do it. So it's better not to be greedy and to target your questions to a smaller number so you can ask them. But in the terms of, in the area of genes that affect nutrient metabolism, there's another factor that we have to be considering. And I'm going to tell you that you have to understand what people have been eating to interpret what the SNP means. Now, how can that be? Well, I think, again, I told you that you might have a SNP that makes you metabolically inefficient. And in the presence of a diet low in that precursor, you might show up as having a problem. That metabolic inefficiency might result in your metabolizing folate less well, and you need more folate to prevent birth defects or something like that. But if you've been eating a diet very high in folate, you might be eating around that inefficiency, and so even though you have it, there may be nothing wrong with you. So if we do a scientific study and says, is this polymorphism, we call them SNPs, is this associated with some kind of dysfunction or disease? For many people, eating a diet high in that nutrient, folic acid, let's say, nothing wrong, and we say, this SNP doesn't do anything. So we have to remember that we have to look for the group that is limited in two ways. They're limited by the quality of their diet, and they're limited by the metabolic inefficiency due to the SNP. And if you don't know both pieces of information in your epidemiological study or your clinical study, you're going to either reject SNPs that are really functionally quite important, but only important if you're eating a poor diet. And I think the fact that diet allows us to eat around these polymorphisms explains why they are so common. I'm going to tell you about some SNPs that more than half of women in North Carolina have. They're bad, cause an inefficiency. How did they survive? Well, it's because in older times, diet was such that it didn't matter because you ate a diet high in that nutrient, so you didn't care if you were a little inefficient. And it's only as we change our diet to remove something like fat from our diet that we start removing some of the nutrient that got us by this SNP, and now it might be more important. So we'll talk more about that later. But the take-home message is if you're going to fund studies in nutrigenetics or nutrigenomics, you have to be collecting diet, phenotype, and the SNP information. So how does this all work practically? Are there real-world examples that I can show you that this could be useful to you? Well, I love choline, and so I'm going to focus around that area, but there are many other areas in folic acid metabolism, in SNPs that make you have cholesterol go up when you eat a high-fat diet versus SNPs that make cholesterol go down, etc. I'm going to focus on the choline ones. You'll remember that this is a nutrient that until 1998 we didn't know was essential for humans. In 1998, the Institute of Medicine set an adequate intake for this nutrient, and that adequate intake may be right. It may be a little low. In some studies that we've done recently, giving the adequate intake, about 10% of subjects still get sick from not having enough choline, and we have to give them a third more to get them by. So that intake is somewhere right, or near right, but might be low. So this nutrient, when we did studies with it, and we were among the first to try to prove humans require the nutrient, so we put them in the hospital and took the nutrient away from them. And what we found was that almost all men, almost all postmenopausal women developed liver or muscle damage when deprived of this nutrient. But among young women, premenopausal women, only about 56% got sick, and 45%, 44% didn't get sick. So if I were only studying women in my clinical study, premenopausal women, I might have been very confused. Some need it, some don't. When you average it all together, depending on the luck of the draw, I might have said choline isn't important. I might have said choline is important. And there was a lot of noise. Half need it, half don't. That's a lot of noise. That's generating a lot of variance in the results. So how do you explain it? Well, it turned out choline is a nutrient that we can eat in foods, and foods like eggs and meats are very high. Some plant sources are much lower. And you can also make it in your liver. And the gene for encoding making the liver is a gene called PEMT, and it methylates one phospholipid to create phosphatidylcholine, which can then release the choline from it. And we said young women don't need to eat as much. The only way they could not need to eat as much is if they could make it. This is the only pathway for making it in humans. So could it be that they have more capacity to make it? And indeed, what we discovered is that this gene has an estrogen response element. That means ahead of the gene, in the regulatory area of the gene, there is a site that responds to estrogen complex and turns the gene on. And it turns out that this gene turns on at the concentrations of estrogen that young women have, and it reaches its maximal turn on at the concentration of estrogen that you achieve in the 3rd trimester of pregnancy. So this gene was designed to be turned on in women who could get pregnant, and turned on even more in women when they were pregnant. And so here you see in the human, over here, the gene is pretty inactive at low estrogen concentrations, and then when you reach about 10 nanomoles per liter, which is the concentration you reach at the 3rd trimester of pregnancy, it's maximally turned on. And if I showed you what estrogen does during pregnancy, you see it rises from 1 to a little over 10 during gestation. So women were designed to have a gene switch in front that's estrogen responsive. Men like myself, and unfortunately postmenopausal women, don't have the key ingredient for turning on this switch, so they don't make a lot of choline. When they're taken out of their diet, they always get sick. And men, young women, may or may not make enough, okay? The ones who can turn on the gene with estrogen don't seem to need it during pregnancy very much. So Nutrigenomics explains why young women might be needing a different amount than men do, nutrigenetics. But the problem we face is, why do 44% of young women still get sick when deprived of choline? Why is it that they have estrogen, perfectly good estrogen? Why are they not making their own choline? And again, if you look, they have a set of polymorphisms in their gene, spelling errors in those genes, in that gene, that happens to be right at the estrogen response element, and they don't have one. It just doesn't respond to estrogen. To show you what that looks like, here are liver cells from human donors, motorcycle riders without helmets, who have the normal gene, and when you give them estrogen, they turn the gene on. Here are donors who have one bad allele and one good allele. And here are the ones who had both alleles be the allele that causes the problem. And so with two copies, they don't respond to estrogen at all. So here we have now another use of nutrigenomics. Now we can not only explain why young women are protected, but why some young women, even though they should be protected, aren't, because they're among the population that has this polymorphism. And here's another way of looking at it. These are the three variants. So GG is the functional type. CC is the one who has two of the effect alleles that block the effect of estrogen. And in women, you can see as you have more, sorry, let me go back. Yes, in women, you can see as you have more alleles, you become more and more likely to develop liver or muscle damage when deprived of choline, and it doesn't make a difference to men. Why doesn't this make any difference to men? Well, they don't have estrogen. Even if they had a good switch, they couldn't turn it on. So they could care less. They're always going to get sick when deprived of choline. So here, it affects only one gender during the time they have loss of estrogen, and it makes a big difference to whether they have to eat this nutrient or not. And when we've learned about gene mutations, when I was in medical school, for instance, they were always rare things like homocystinuria or phenylketonuria. But here we're talking about 74% of Caucasian women in North Carolina have one variant allele, and 22% were homozygous. So not rare at all. In this room, a quarter of you, of young women, or everybody here, has that SNP, if you're Caucasian. I'll talk to you more about how it differs among populations in a minute. So again, I said that we have SNPs causing a metabolic inefficiency. In this case, it turned off your ability to make the nutrient yourself, and that's your inefficiency. And we went on and found that there were a number of polymorphisms that people who became sick when we deprived them, fed them a low choline diet, had. And they were in folate metabolism. This gene could increase your chances of becoming sick 85-fold if you were a man or a postmenopausal woman. And that gene controls the use of folate as a methyl donor. It's the switch that sends folate either to make DNA or as a methyl donor. When it has its polymorphism, it tends to not send it as a methyl donor. Choline has to make up for that by being the replacement methyl donor, and so it becomes a big use of choline. There's a SNP in the conversion of choline to betaine, and that increases your risk 20-fold. And I'm going to come back to this gene because I've been neglecting men in this story, and I'm going to come back to you at the end and make you interested in what I'm saying as well. So at least 3 different polymorphisms in pathways of choline metabolism dramatically change whether you're going to require it or not. So imagine I'd done my first study, I didn't know any of this, I would have had some people getting very sick, some people not caring at all, a big fuzzy data typical of nutrition data, and we would have had long arguments in the literature, is this real or is this nonsense or whatever. But doing the genetics, you can go back and say, aha, well, every responder looks like this, every non-responder looks like this, I can predict with almost perfect accuracy who's going to be a responder, non-responder. I now have a useful clinical study. I don't have to stick it in the trash bin and pay to do it again. I can tell people what to expect. So the folate metabolism, when I talked about this SNP, increased risks in populations by 7 to 85 times. And again, I told you what it does. This is the choline dehydrogenase SNP. Again, it's the conversion of choline to betaine, which is then the methyl donor. And I'll come back to that in a minute. Now, the other thing I wanted to say is SNPs, polymorphisms, are not distributed equally around the world. We inherited them from our ancient ancestors. And our ancient ancestors differ by where we come from and where our, what population we exist in. So here I went to the Gambia, Africa, and there I found that their dietary intake of choline was exceptionally low, about a third of what we think is the adequate intake. And so I was saying, boy, if they have the polymorphisms that make people inefficient, they're going to be really in big trouble. But lo and behold, when I look in Africa, here's that gene for turning PEMT on. It's missing in Africa in homozygous form. Very few women have it. If I look at the choline dehydrogenase SNP that increases requirement, again, missing in Africa. And methyl tetrahydrofolate dehydrogenase, missing in Africa. I suggest to you that the reason it persists, it's a bad SNP, but it persists in people of European background is your mothers fed you eggs when you were a young child in Europe or in Caucasian America until 1970 when we told people fat was bad for you. And so everybody ate eggs every day, pretty much. Mother wasn't doing a good job if she didn't send her kid out with an egg breakfast, right? So you ate enough choline, you didn't care about the SNP, it survived. In Africa, where the intake is extremely low, if you had this SNP, you probably got selected against, and over generations, thousands of years, those SNPs have disappeared. So what these polymorphisms do and where they're going to be important is going to vary around the world, and you're going to need to know the population you're studying and do genetic testing to figure it out. You can't assume the distribution. So let me go on. Let me say that when people are eating a low choline diet, some develop liver disease, most of them, but some develop muscle damage. And we've been puzzling, why is that? Why do some people have muscle damage? Now, the liver damage comes because to package VLDL and secrete fat from liver, you need choline. And we showed that we could predict who would get fatty liver by a pattern of these SNPs that I've been talking about in choline metabolism. This is the group that has low fatty liver. This is the group that has high fatty liver among 850 people with liver biopsy proven fatty liver. And we can see that the SNP distribution is very different between the two groups. And we published a paper saying we could predict whether you were going to get fatty liver as your BMI rose because you had problems in packaging VLDL because you didn't have enough choline because of these pathways. Now, I told you some people get muscle damage. Why? And we're just publishing a paper that says all of the people who present with muscle damage, the 10% of the population in our study, all had a SNP in the transporter for choline that takes choline into the mitochondria. If you have that SNP, you're going to present not with liver disease, but with muscle disease, because something about mitochondrial function and choline is important for muscle. And how do these people present? With very high creatinine kinases, CPKs, in the 20, 30,000 when deprived of choline. And that's not an unusual thing to see. Even the military is complaining that they have a number of troops that they can't force to exercise because as soon as they do, their CPK rises. Perhaps they fall into this group. But in any event, knowing something about genetics helps us figure out what's going on. In my last 3 minutes, I just want to make the men happy. I told you how it matters to women. How does it matter to men? This, by the way, is in Seattle, how they take sperm from the sperm bank to the clinics by bicycle. It's very green. So in any event, this SNP that I told you about that increases your requirement for choline, when we took it, our technique has always been, once we find something in the human, go make a mouse without the gene and just ask what else does it do, because we might not have been smart enough to look for it in the human. And so when we knock this out of the mouse, and this is the gene that we knock out of the mouse, it's in the mitochondria, this gene, we found that the male mouse that was homozygous for this knockout had sperm that just didn't move. Couldn't swim. Of course, I didn't think to study that in the humans that I was making choline deficient, because we normally don't show them movies and get sperm from them. But their mitochondria looked really terrible. So this is in the knockout mitochondria in the sperm. You can see very empty. They have terrible looking mitochondria compared to the normal mitochondria here in the wild types. So they have terrible mitochondria, and they can't make ATP because of something wrong in choline metabolism. They don't make ATP. You can see quite a difference in ATP generation. And we said, what if we give the product of that gene, that pathway, betaine, which we've blocked because choline can't be converted, and we could restore most of that ATP difference. So mitochondria needs this pathway to make betaine for some unknown reason that we're still working on to make sperm be able to make energy. So you say, okay, mice do this. I'm not worried about the mouse population's fertility. Is this true in humans? So again, we went back to men, and we found that in our population, between 5 and 9% of men have a SNP in this gene, choline dehydrogenase. That's the numbering in the SNP. That's just an identification system so other people can figure out what you're talking about. And 5 to 10% have that SNP. So we collected some of those men, and we looked at their mitochondria and their ATP production. So here is good gene, half and half, bad gene. Mitochondria look terrible again in these men. ATP looks terrible in these men. Slightly different from the mouse. Instead of not moving at all, their sperm swim in circles. Still not very good for accomplishing the mission of a sperm. And so these men have a fertility problem because their sperm swim in circles, because they don't make enough ATP. And why in mice they don't move at all, and in men they swim in circles, I don't understand yet, but mice and men are different, we know that. But again, it may be that betaine, the nutrient found in the roots of plants like beets and spinach, might be very effective at treating this form of infertility. 5 to 9% of men are homozygotes, so that could be 1 million men, 2 million men, who knows? So in any event, understanding genetics helps you to figure out who is a responder and a non-responder. Most men's sperm could care less about betaine. This population may care a lot. So I think nutrigenomics is important for identifying responders from non-responders. If you start using precision nutrition, it should reduce the noise in your research studies. Those of you doing clinical trials should be collecting this kind of information, because even if you don't analyze it, until after you find out you have a gray result with 85% doing nothing and 15% responding, you can go back, if you've collected it, and then ask, what was special about those 15% responders? How were they different from the 85% who did nothing? You can't ask too many questions, so don't get greedy, because if you ask too many questions, you'll never have the statistical power. But if you do a study with about 100 people in it, you can ask about 300 polymorphisms and still have adequate power if the size of the effect is reasonably good. If you ask 2 million questions, you've got to have 1 million, 2 million, 3 million people in your study. It's never going to happen. But I think you should collect diet information because, as I said, fighting the SNP and seeing it did nothing means nothing if the person was eating a lot of betaine in my men study, for instance. Those men may have nice swimming sperm, even though they have the polymorphism, so I have to measure both. And finally, I think that this will become the foundation for what eventually we'll call personalized nutrition. I think there'll be diagnostic tests for, let's say, a woman who has this SNP and has to be more careful during pregnancy. She may need more choline during pregnancy. There may be a test that an OB does to tell her to be more careful. I think for most, we'll say, don't bother with the tests. We know 1/3 of the Caucasian population has this. If you're a Caucasian, you probably need to take some insurance out, eat something that contains this because you have a high probability of falling into this. And maybe in the days of 23andMe and other genotyping things, it'll be so common that everybody will know their genotype, and then we'll have to have some kind of computer program that tells them which nutrients are they at risk for needing more of based on a catalog of SNPs that we develop in the next few years. I'd say there's hundreds now. Probably you need thousands to make this a useful endeavor in terms of sequencing people's DNA. But that's the story and how you could use it. This work was done by a bevy of graduate students and postdocs and young assistant professors in my laboratory. I want to thank all of them, and the USDA and the NIH and a number of foundations for funding the research I described to you. And whoops, I'm gone past my story to the next one. But thank you for listening, and I'll answer any questions.